Good morning, everyone. So it's my pleasure to introduce Marissa Baskett this morning. And we're changing gears a little bit. We've heard a lot of molecular biology so far, and today we're going to hear a fabulous talk on ecology. Marissa was trained at Stanford for her undergraduate and then at Princeton for her PhD work, and if you put together Princeton and ecology, then you automatically come up with Cy Levin, who of course was her mentor. And the other thing about Simon is that he was the past president of the Society of Math Biology, and we certainly wish he was here, but he's a busy guy. And so Marissa's work has been very impactful in studying the ecology of, I just blanked out this morning. Sorry, Marissa, I haven't had my coffee yet. And but what's amazing about her work is that she's able to pair together actual application to the theory that she studies, and that's what we'll see coming here today. And also, I just want you to think a little bit about the ideas that we can translate lessons learned in different areas to the work that you're doing. So even though she's talking about ecology, there's probably some important lessons that can be translated to what we're doing today. I also understand she's an avid scuba diver, so maybe we'll see some cool underwater pictures today. So anyway, without further ado, Marissa, welcome.
Thank you very much for the introduction and for the opportunity to speak here. This has been a real honor, and it's been wonderful to be able to join in on this meeting. So today's talk really starts with this issue that we are in this era of unprecedented global environmental change. And that's coming from a variety of human influences, first and foremost of which is climate change. And with climate change, it is a true global issue such that emissions in one location affect the potential climate everywhere on this planet, which leads to a lot of uncertainty in terms of what the future might look like, as illustrated by these different projections put forward by the Intergovernmental Panel on Climate Change, everywhere from those blue lines about saying we do everything we can to reduce greenhouse gas emissions now, to that red line of business as usual in terms of greenhouse gas emissions, future greenhouse gas emissions, which leads to very different future climates. And raises the question for a local manager, say of an individual coral reef somewhere, asking what can they do? They cannot necessarily affect change in global greenhouse gas emissions. So what options are available to them in the face of this change? And a lot of talk has been going around of, well, let's then focus on protecting the capacity for that local system to respond to global change. So protecting response capacity can mean a variety of things depending on what ecological scale you're focused on. If you're focused on the genetic scale, then you might think of it in terms of protecting the potential for genetic adaptation. And so this is in terms of microevolutionary change, changes in gene frequencies within populations that can occur quite rapidly. And we've seen genetic evolution in response to climate change, the evidence for that in some organisms, primarily in phenological traits. And so that raises the question of, well, what drives that adaptive capacity and can we protect it? On the population level, we think of that in terms of what drives population persistence into the future. And in the community level, the question is then what drives the maintenance of a particular community state, and can we protect those characteristics? So I'm going to step through examples of how we might think of what the drivers are for response capacity on each of these ecological scales, which can then inform priorities for management decision making. And as my sample system, I'm going to choose coral reef systems, because corals are one of the most vulnerable taxa to climate change. They are a taxa that are living on the edge. And this comes from the basic coral biology that they are many organisms in one. There is a coral animal, that is the invertebrate that builds the structure you see. Inside of it is a symbiotic algae, a plant that photosynthesizes. And that photosynthesis creates the food for the coral as well as the algae. The coral provides habitat and nutrients to those symbionts. And together they form this holobiont. But when that holobiont is stressed, the coral loses its symbionts. And because the symbionts have the photosynthetic pigments that give the coral its color, the coral just is left with its calcium skeleton then and looks white. So we call that bleaching. And when that stressor is extreme or prolonged, then that is fatal to corals. Now, many different stressors can lead to bleaching. And before the late 1970s, bleaching was considered this kind of odd physiological rare thing that happened just a little bit here and there sometimes in a weird event. But since the late 1970s, we've seen this increase in incidence of what are called mass bleaching events. That's multiple coral provenances experiencing large-scale bleaching throughout the Earth's tropical oceans. And those have been associated with particularly high temperatures in El Nino years. And this particular plot, because temperature, extreme temperature, whether it's extreme high or extreme low, is one of those stressors that can lead to bleaching. Now, this plot happens to stop in 1999 because of the paper it comes from, but we just had last year a mass bleaching event associated with last year's El Nino, and it hit the Great Barrier Reef particularly hard, and that is one of our most pristine and well-protected reefs. We're still getting a sense of what the level of mortality from that event is. Now, increasing temperatures and the stress associated with that is not the only way in which greenhouse gas emissions are affecting corals. Carbon dioxide emissions in particular are leading to what we call the other CO2 problem, ocean acidification. And so this has nothing to do with climate, everything to do with simple increases in concentration of carbon dioxide in the atmosphere means some of that diffuses into the ocean. The oceans are actually a major carbon sink right now, and that carbon dioxide then interacts with water and hydrogen ions to eventually become carbonic acid. And so the pH of the oceans, which is remaining within the basic range, becomes a bit lower, a little bit more acidic within that range. So that's what we call ocean acidification. But it's not the pH that's the issue. It's actually that carbonic acid. That interferes with the ability for marine organisms to build calcium shells and skeletons. It becomes harder to build up that calcium. And so with that, corals, of course, rely on building on these calcium skeletons in order to build that fundamental structure that forms the basis of coral reefs. So with that, we have here on the x-axis the idea that we're getting changes in carbonate ion concentration, and the y-axis changes in temperature, two major stressors on coral reefs. We can look at what corals have experienced through the past glacial and interglacial cycle and see that today already falls outside that past experience. Then the question becomes, as we look into the future, might we be crossing thresholds for corals' ability to build their calcium skeletons and thresholds of the temperature stress that they can withstand? The estimation is that on average, with about 1 to 2 degrees above the typical summer maximum, that's when you see bleaching. And so will bleaching become an annual event, which would be catastrophic to the future of coral reefs? So a lot of the coral world is very worried about, are we going to cross these thresholds? But the other thing we know that is highly characteristic of coral reefs is their diversity. They blow any other system out of the water in terms of the diversity that we see in this system. This is a picture I took from the Great Barrier Reef about a year before that mass bleaching event. And you could just see, with a single glance, the morphological diversity of corals right there in one snapshot, right? And that, beyond that are so many other types of diversity going on, so many different coral species, so much genetic and phenotypic diversity within each species. And with diversity, that means we are not living in a world of one single threshold. And diversity really, from the genetic up to the community level, it should be what we fundamentally expect to underlie response capacity. So let's look into what role that diversity might play, what might drive response capacity across each of these levels. For the genetic level, I'm going to focus on potential adaptation to ocean acidification. And then for the population and community level, I'm going to focus on thermal stress. But note where that might interact with ocean acidification. For this first of the three pieces, my collaborator is Joshua Maiden at Macquarie University in Sydney. And before I launch into genetic adaptation to ocean acidification, I do want to say that some of my older work, which just was a bit old to present today, did look at genetic adaptation and thermal tolerance for those symbionts that live within the coral. And we did find that evolution, rapid changes in gene frequency within populations, this microevolution, can occur rapidly enough to play a role in future coral persistence under climate change, but it depended on the climate scenario, how fast that climate change was. And if we looked at what really drove that adaptive capacity, diversity really showed up, of course, that's what we expect, but survivorship showed up as a key demographic driver compared to other potential demographic processes. Such as just a quick synopsis of some older results on adaptation to thermal stress before we get to adaptation to ocean acidification. So we asked an analogous set of questions for ocean acidification. Do we expect evolution to play a role in persistence into the future? And if we have this model of coupled evolutionary demographic dynamics, what traits are most likely to influence future persistence? What drives that response capacity? Ergo, how do we use that in conservation prioritization? So to look at this question, we built on an existing integral projection model that Josh Maiden had already made without any evolution and extended it to add evolution. And so an integral projection model is a case of an integral difference model where we take the population distribution over some characteristic at one point in time step, multiply it by the growth, survivorship, and reproduction as a function of that characteristic to map that to a distribution in the next time step. And the characteristic we chose is size because in corals, size is highly influential on these demographic processes of growth, survivorship, and reproduction. Joshua carefully built this model based on an amazing data set that he developed for this acroporid coral. So this is a tabular coral. For its morphology, it grows up into this tabular form. He chose this coral as his focal species because it's abundant throughout the Indo-Pacific, which is the most diverse area of coral reefs. It provides habitat structure, and it's a potential community dominant. It has this very important role to play in the community. It's very productive, but also it's vulnerable to dislodgement under wave stress. Think of this tabular coral, right, that with that base, you could think of that being quite fragile to a wave coming through. And if what ocean acidification does is weaken your skeleton, it's going to interact synergistically with that wave stress by weakening that stronghold. So here's a community dominant that we think is particularly susceptible to ocean acidification effects. So to look at how Josh built this model, first starting with the growth equation, here is where the real hard work of building an IPM is. Tagging individual corals, here at different depths because growth rate depends on depth, and following them through time in order to measure what are your growth rates. From that, we can come up with a relationship for a size at one point in time. What are the distribution of sizes at the next point in time that we can expect, so we can have a probabilistic description of this. Next, for survivorship, that size-dependent survivorship, here's where that dislodgement from wave stress comes in. And that dislodgement likelihood depends on the shape of the coral. So first we need to translate size to a shape factor, which is going to be a distribution. Not every size is exactly one shape. Again, we get a probabilistic description of this. Then we compare that factor to a threshold that leads to dislodgement under a particular level of wave stress. And here is where Josh's biomechanics background really comes in. This is based on a very mechanistic understanding of the limiting strength of that attachment, as well as using data on what the potential wave stresses are, right? That's going to vary year to year. Storm levels are going to vary year to year. And so we use then a probabilistic description of what the potential wave stresses are to then get to a probability of survival, as it depends on that shape factor. So the likelihood of dislodgement, of mortality due to dislodgement, which is the main source of mortality in these corals, is based on size, and therefore because of that shape factor. But we also put in a simple baseline survivorship just due to natural senescence that's size independent. Finally, because this is an abundant community-dominant coral, it is a well-studied coral, and we luckily already had data on what the reproduction relationship with size is. So after a particular size at maturity, we get this increasing reproductive output with increasing size. And we just put that relationship into our kernel. So this is the model that Josh had already built. The next step that we wanted to take is let's add evolution to this. And if you think about, with increasing ocean acidification, it becoming harder to build a calcium shell with the same energy, you basically have two choices, two extremes of choices. You could either grow slower and maintain the same skeletal density, or grow at the same rate, but then you'd have a weaker skeleton. Or you could be anywhere in between. So we model this as a quantitative genetic trait. Our assumption is many loci contribute additively to this trait, as well as having potential random environmental influences. So it can take on a continuum of values, as compared to, say, a Mendelian trait that's just one locus, multiple alleles. And so in order to have this trait, which is this, where does the investment in calcification go toward growth or skeletal density, we need to know something about how the carbonic acid concentrations in the ocean translate into the calcification potential. And so there are existing relationships out there. We picked the most optimistic scenario of those of our choices. And then we also needed to be able to translate density into that strength of being able for the hold fast to hold on, right, to get to that size-based survivorship. And we assume that it's only the coral strength that's changing, not the substrate, which is, again, a more optimistic assumption. The corals need all the help they can get right now, so keep in mind that we do have these two optimistic assumptions. We do test the effect of these assumptions. So to put in our evolutionary change, we have to make two more key assumptions. One, we have to get to a fitness as a function of this trait, some sort of fitness metric. So for that, we take our IPM and we assume a stable size distribution, which from then we can just use, take the discretized version of the IPM, the leading eigenvalue. That gives you the population growth factor for a particular phenotype. And that's exactly our fitness. So we're assuming rapid change in size distribution relative to evolutionary change here. So from that, we get individual fitness as a function of phenotype. And then we want to know the change in phenotype through time as ocean acidification changes. So we use a fundamental quantitative genetic model, the Breeder's equation model, which just looks at the change in mean phenotype from one time step to the next as it depends on what we call the selection differential and the heritability. So another key assumption here is that the phenotypic distribution is normally distributed with a constant genetic and phenotypic variance. So it's only the mean that's changing. So from that, you can integrate the fitness over a given phenotypic distribution with a given mean, and that gives you the mean population fitness for that genetic mean. And there are two key differences between that mean population fitness and the individual fitness. One is because that individual fitness function is asymmetric, the peak is a little bit shifted. You don't want to be exactly on the individual fitness peak because then half your population is poorly adapted. So you shift a bit away from there. Second is because you have this variance, not everybody can be perfectly adapted. And so the maximum population growth rate is always going to be a little bit lower than the maximum individual growth rate. So from this, you basically take the derivative of this guy, normalize to get your selection differential, multiply that by the proportion of the variance that's heritable, that has a genetic component, as opposed to a random environmental component, then you get your change in mean trait. So to look at our two questions, the question is, does evolution matter? We can ask, well, does your mean population fitness stay above 1? Which is basically, does your population stay above self-replacement? More than one individual in the next generation for every individual in this generation for different levels of heritability. Heritability of 0 means that you have no trait change, right? No evolutionary change, so we can compare that to increasing heritability, more and more that variation being genetically driven such that you can get faster evolutionary change. And then for our question of the role of different traits, here we do a sensitivity analysis. And the approach we use is a global sensitivity analysis, where essentially you specify a range for every parameter, and you do thousands of simulations where for each simulation you do a random draw for all parameters. And from all those thousands of simulations, you form a series of regressions, and through those regressions you can get importance values for all of the different parameters in terms of their relative influence on the outcome. In this case, what's the population growth rate at the end of the simulation? So here we have, for the role of evolution, we have mean fitness as a function of time, this mean population fitness, for 4 different future climate scenarios, ranging in the upper left that everybody does everything they can now to reduce greenhouse gas emissions, to the lower right of business as usual. The different lines here are different levels of heritability. So what we're seeing here is evolution, in this case, evolution toward increasing investment in skeletal density as compared to growth, leading to changes. And in 3 of these 4 graphs, no matter what happens, whether or not you have evolution, we get our population growth factor staying above 1, staying above self-replacement. Remember, we did have some other optimistic assumptions in there. It's only in this last case, this business as usual case, where we get the potential for lack of population persistence under future ocean acidification. And what we see here with evolutionary change is that we do get a shift in how we do see later that this decline below population persistence happens later. There's a little bit of a delay there. But it's not much. Evolution is not making a difference in terms of whether overall persistence can happen. One thing to note here is that if we were to plot that maximum individual fitness, we will get persistence into the future. So in a world where this particular trait is 100% plastic and adaptively plastic, every individual can respond to an environmental cue that tells them exactly the right thing to do, then we could get population persistence. That adaptive plasticity is not something we had in the base model. So then, looking at our global sensitivity analysis and the role of these different traits, what we see first and foremost is this reinforcement that in this particular case, evolution is not mattering as much, which is in contrast to what we found for the thermal stress model. These orange cases, phenotypic variance, heritability, these genetic traits aren't really driving the future outcome. Really what's driving it is background survivorship, skeletal density, and as a third, recruitment. These ends as distant fourth growth. So these demographic processes. And I should say this is the global sensitivity analysis for this last case where we might predict the potential for lack of persistence. So what this tells us, in addition to saying that survivorship is important to the future, which itself is not that surprising, but it is surprising how it might outweigh other factors. The other thing it tells us is that we focused on evolutionary change within this calcification process, assuming the energy devoted to that process is constrained. But of course, evolution can happen in other ways. We have resources devoted to multiple processes in the organism spanning across survivorship, growth, and reproduction. And if there's a heritable basis to how resources might be devoted to, say, growth versus reproduction versus survivorship, evolutionary change in that kind of investment will likely have a greater effect on future coral persistence than evolutionary change within that calcification process. So empirically now what we need to do is understand what is the heritable variation, is there adaptive capacity in that resource investment across life history traits. All right, so that's our genetic adaptation model. Now we're going to shift gears to thermal stress and population persistence questions. My collaborators for the next two pieces are Nick Fabina. He worked on this when he was a PhD student at UC Davis, and Kevin Gross, who was at North Carolina State University. So, on the population level, we're asking about future persistence. And in particular, we're wondering, okay, what coral traits most drive future persistence, and what environmental conditions most affect future persistence? Because this can inform priorities in terms of who to protect and where to protect. Who is most vulnerable versus who is most likely to make it into the future. Now, whether or not you protect the most vulnerable or the most likely to make it into the future depends on your risk assessment and your worldview as a manager. You might argue that you should protect the most vulnerable because those are the ones that are going to need the most help. You might argue that you should protect the most likely to persist because those are the ones that can make it through and be our source populations for future coral reefs. And so that depends on your view of the future. In particular, this optimal control analysis by Eddie Game and colleague said that you might want to protect the most vulnerable when you have low disturbance frequencies and the most likely to persist, the strong, when you have high disturbance frequencies. Either way, knowing where and who is most vulnerable and most likely to persist can inform management priorities. So to look at this question, we extended an existing model that Alan Hastings helped build with Pete Edmonds and Pete Mumby and Helen Edwards. Pete Edmonds is another coral ecologist, so it gets a little mixed up. Where they're looking at coral competition with macroalgae for free space. So free space here is anything but corals and macroalgae. Both of them can recruit to that free space. So we divide out coral recruits from coral adults so we can understand relative influences of recruitment versus growth. So we have coral recruitment to free space, maturation to adults. What we're following is the area covered by each of these categories. Corals then grow to occupy more and more of that space, and both of these experience senescence, natural mortality, to free up more space. Macroalgae also grow into this space, and the reason we focus on including macroalgae is it also has the capacity to overgrow coral. Both directly as a shada, it also releases chemicals that can negatively affect neighboring corals. That overgrowth happens on both recruits and adults, and corals cannot recruit to places where macroalgae is. They can only recruit to that free space. But macroalgae experience herbivory, which then frees up space, and the more corals you have, the more concentrated that herbivory gets on the macroalgae. So there's feedback here, a nonlinear feedback of corals onto increasing macroalgae mortality. And we also extend this model to include multiple patches with dispersal between patches. So that gives us our base dynamics, but we want to add thermal stress to this, not just as increasing mortality, but as a stochastic event, so we can look at what happens when you have different locations with different levels of thermal stress. So to do this, we use a metric developed. Originally, Simon Donner was a big part of developing this. We got Cheryl Logan then continued to extend this, where you use degree heating months. So remember I said if the temperature or stress is extreme or prolonged, that's fatal to the corals. So the degree heating month encapsulates both. It's the number of degrees times the number of months that the mean temperature in your summer is greater than the typical summer average. And that's been a very good predictor of bleaching likelihood. And so we use data, global data of degree heating months for the latest IPCC assessment report developed by Cheryl and Logan and colleagues. And so we take that degree heating month and we translate that into a survivorship probability. So if the degree heating months are low, you get no mortality from bleaching. If they're intermediate, we get partial bleaching and partial mortality. And if they're high enough, then you can get full fatal events for these corals. So we use basically two thresholds with a linear relationship in between. So we implement this as a pulse-impulsive or semi-discrete model. We run the deterministic ODE for a year and then do a random draw of degree heating months to determine if and how much mortality occurs from bleaching as a one-time stochastic event. And then we just repeat that through for many years with a burn-in and then have a run after that. For this question of what coral traits drive future persistence, we again do a global sensitivity analysis. For what environmental conditions most affect future persistence, we focus on the question of are we more worried about increasing frequency of bleaching events or increasing magnitude of bleaching events? Because different oceanographic characteristics in different locations will affect which of those you expect. So starting with this magnitude versus frequency question, on the x-axis here we have frequency, on the y-axis we have magnitude. We have two metrics we look at here that basically tell us the same story. On the left, this probability of coral persistence. On the right, the proportion of mean coral cover. And so if we increase the frequency of disturbance, we see some effect on future coral persistence and cover. But it's a lot smaller than the effect compared to increasing magnitude of these future bleaching events. So what really affects this capacity for corals to continue to dominate tropical rocky reefs is this question of what are the magnitude of these future bleaching events going to be. And so we can then identify, okay, what locations do we expect to have future high and low magnitude? For the coral traits, this is one sample future climate scenario. Across all the future climate scenarios, we found 3 traits consistently fell out as the most important. Adult coral mortality, that's that baseline natural mortality, coral growth, and bleaching resistance. And again, it's not surprising that these are important to the future. What might be surprising is that how much they outweigh other traits that you also might think would be important, like coral recruitment and dispersal between locations that might be experiencing different thermal stress. And so in particular, for a coral to persist into the future, it needs at least 2 of 3 things. And that's low mortality, fast growth, and high bleaching resistance. But there are trade-offs, life history trade-offs, in the ability for an organism to achieve these things. And a key thing to note here is that 2 of them are things that we expect ocean acidification to strongly influence. That survivorship, remember the dislodgement likelihood, and the coral growth. And so there is really a potential for a synergistic interaction between ocean acidification and thermal stress here. So we expect life history trade-offs between these 3 things. And in particular, the coral morphologies, in addition to that tabular morphology I mentioned earlier, very common coral morphologies are this mounding morphology, which is a very slow-growing coral, but has low mortality. You can imagine this thing can take a beating from waves and not be dislodged in the same way as that tabular coral, and also high bleaching resistance. Because the bleaching is actually a temperature by UV interaction. And so with the mounding morphology, the symbiodinium can retreat to the interior of the coral and be shaded and be more likely to survive through, and then after the bleaching event occurs, regrow back out from there. And so those do have checked 2 of those 3 boxes then, as compared to our branching type morphologies, which are super fast growing compared to these mounding corals, 10 times as fast typically. But as you can imagine, very more brittle, more easily to break, experience high mortality, and with all that exposure are more susceptible to bleaching. And so because of this, a lot of empirical studies go out and categorize winners and losers and say, well, the mounding corals, the massive corals, are typically more of our winners into the future. When you see a bleaching event, those are the ones that don't bleach or survive afterward. And so that raises the question of, should we be focusing our conservation priorities on them? We do expect a high disturbance future, and if we're going to choose between, as this Cote and Darling paper put it, ecosystem resilience and ecosystem recovery, what we really should focus on is this resistance. Let's focus on these guys that are going to be most likely to make it into the future. But thinking about that as a single population at a time, even if it is thinking about different populations and the diversity of them, still to me forgets something fundamental to these corals. That's not just that their diversity, but their interaction with each other. Right? All these corals are interacting together, coexisting somehow, this extraordinary diversity. And the overall potential to maintain a community-dominated state is not just the sum of each individual response. Each individual population is responding but interacting with the other populations, and that interaction drives the larger overall response. They are a complex adaptive system. And anybody who studies complex system theory knows that diversity should be a major driver of the capacity for a system to respond to change. And so that led us to this idea of, let's look at this at more on a community level. And so then, from a complex adaptive system perspective, it's thinking about the system as being able to be in different states. And what maintains the target state, the coral-dominated state. So we're going to focus now on this idea that you have these two broad categories of types, your resistant type and your resilient type. And that idea doesn't just apply to coral systems. We could apply this to lots of systems. For example, in forests, you have your slow-growing trees that are pretty resistant to disturbances like fire and drought compared to faster-growing, higher mortality trees that then burn up more quickly or are more susceptible to drought. There are many systems you could talk about these different types. And these types interact. There's the obvious direct negative interaction. They are competing for space. But there is this indirect positive. They are competing with other species for space too, other taxa for space, like macroalgae. And so if one coral can keep away macroalgae, again through that concentration of herbivory during a bleaching event, maybe that can be an indirect benefit to another coral. You might have this enemy of my enemy is my friend effect. So if you have a direct negative and a potential for an indirect positive, the question is, which one then predominates in the overall outcome? Do you get enhanced or lowered community level resilience? Well, if we're going to use that word, resilience, we have to be careful about the definition. And so we use the Halling definition, and that comes from the idea that an ecosystem can be in multiple states, like a coral-dominated state and a macroalgal-dominated state. And there might be some conditions for which both states are locally stable, and you have an unstable threshold in between them, which we could represent through a bifurcation diagram or through this ball and cup diagram. And the question of resilience then is, given a disturbance, what is your likelihood, a pulse disturbance, a one-time short-term disturbance, what is your likelihood of crossing that threshold? What is your likelihood of switching states versus staying in the same state? So you could think of resilience then as the basin of attraction to the coral-dominated state, or and so where that threshold is, or if you're thinking in a ball and cup world, is the height of the hill that the ball would have to climb to get into a different cup. Now, whether or not coral reefs, macroalgal-dominated states, alternative stable states, is highly debated and controversial in the literature. Currently, there's more evidence empirically that this is the case in the Caribbean compared to the Indo-Pacific. We're going to set the question of whether or not it is the case aside and say, if it is the case, what leads to a greater likelihood of maintaining the coral-dominated state? So we have our two different coral types, a resistant coral and a resilient coral, competing with macroalgae. Again, we're doing this Mumby et al. Type model where we're following the proportion of area covered by each type. With the corals then increase their area covered by recruitment and growth, both. So for the recruitment, we analyze 3 different cases. One is just closed recruitment. There's an assumption here that any surrounding reefs have analogous reproductive output and cover that drives that reproductive output toward the focal reef. Having some just low level of external recruitment, maybe there's a source population somewhere else, or having a 2-patch system with different levels of disturbance between the patches. Our default assumption is that little small level of external recruitment. These guys have this natural senescence, this natural mortality. The macroalgae also grow into free space, potentially with external recruitment. And we have competition between the corals, where we analyze 4 different competitive assumptions. One is just that a coral is a coral compared to other corals, regardless of whether it's the resistant or the resilient type. Interspecific and intraspecific competition are the same. The next is that maybe these guys occupy slightly different niches. We do know that they typically they have different depth gradients, that maybe they can coexist by the branching one being a little bit higher than the massive one. Both can get sunlight. So maybe your interspecific is a little less than your intraspecific. And we know they can coexist. Something drives their coexistence. And so that's our default assumption. The next is we could assume one or the other is the superior competitor. And the resilient coral might be superior by overgrowing and overtopping and overshading. The resistant coral might be superior because corals have these stinging cells that they release if they run into a neighboring coral, and they send them out, and that kills the edge, the boundary along those 2 corals. And there's some evidence that these mounding types are a little more effective at putting out those stinging cells compared to the branching types. So that's our competition. We also, of course, have this potential for macroalgae to overgrow the corals and the corals to concentrate herbivory on the macroalgae such that more coral means higher algae mortality from herbivory. So the question then is, because our main extension to this model is adding these 2 types of corals, how do we define them? How do we define a resistant type and a resilient type in this model? Well, the resistance is pretty straightforward. It's bleaching resistance in our particular model. So we, again, have this idea. We take this thermal stress metric, degree heating months, look at the mortality from that. And the resistance is the slope of this value. So a shallower slope means a later level of thermal stress at which you start to see bleaching, which is definitely the case for branching versus massive corals, and a slower increase in bleaching mortality as your stress metric increases. Resilience, again, we can have multiple definitions too. We can think of it as this area basin of attraction to a coral-dominated state. So if we have our macroalgal-dominated state and our coral-dominated state, it's okay what proportion of initial conditions then lead you to your coral-dominated state. That's your ecological resilience. There's also the idea of engineering resilience, right? What's your rate of return to a particular state given a disturbance that didn't knock you out of your basin of attraction? So we can measure this with stability, basically, with the leading eigenvalue of the Jacobian matrix for 1 coral at a time, and the macroalgae, right? Because that's your return rate near your coral-dominated equilibrium. And those 2 metrics generally agree with each other, regardless of which one you do. If you change the coral life history parameters, you'll get increases or decreases in both together. And so for the resilient coral, we give it high resilience by giving it fast growth and having it have a lower rate of overgrowth by macroalgae, which we assume also comes from that fast growth. Versus the resistant coral is low in both of these values, slow growth, and that slow growth leading to faster overgrowth by the macroalgae, whereas we keep the mortality analogous between these corals. And so from that, if we just look at the resistant coral with the macroalgae, we get a very small basin of attraction to the coral-dominated state, versus just the resilient coral with the macroalgae, we get a large basin of attraction to that coral-dominated state. So again, lots of stochastic simulations where we do this semi-discrete or pulse-impulsive numerical approach, simulate the deterministic dynamics for a year, then put in our bleaching event. We initialize near coral-dominated equilibrium, do a burn-in, and then look at the stochastic outcome over a set of years for 5,000 runs to get a full sense of the distribution. And we compare just the resistant type in macroalgae, just the resilient type in macroalgae, versus both together. To really look at this question of when do the two corals benefit each other versus when are they a detriment to each other. And we look at that under different disturbance levels, recruitment dynamics, and competitive interactions. So to orient you to these graphs, we have coral cover. For in blue is always the resistant type, in green is always the resilient type. The darker colors are for each type when the other one's around. The lighter is when each is alone. The black is the total cover when both are together. I'll step you through it. So this is looking at coral cover. Remember we said ecological resilience is the probability of maintaining a coral-dominated state. And so we also look at the probability of that, and it follows these cover dynamics. The cover ones just provide a better illustration. So here we have our increasing levels of disturbance. And we can see at low levels of disturbance, we get declines in each coral cover when the other one's around, compared to if each is alone. Because here you have this negative effect of competition predominating. Total coral cover is higher, though, with both together, because we have this assumption that of niche differentiation. Interspecific competition is lower than intraspecific competition. So low levels of disturbance, you get the detriment of both together for individual but not total coral cover. High levels of disturbance, though, each coral actually has higher and less variable cover when the other one's around, and the total coral cover is higher compared to having each alone. And so it's at these high levels of disturbance that you get that indirect positive predominating. Each coral holding off macroalgae in different ways. The resistant coral by staying there, the resilient coral by growing in quickly, each of them benefiting each other then in terms of holding off macroalgae to benefit their cover as well as the total. And what's particularly interesting here is that the resilient coral benefits the resistant coral in exactly the simulations where if the resilient were there alone, it would be gone. Right? So this is a world where you would predict the resilient can't make it, let's give up on it. That's when it has the most positive role to play in community-level persistence. Focusing on this intermediate high disturbance, looking at our recruitment dynamics, here we have no external recruitment in one patch. Resilient coral collapses. The resistant coral still gets a legacy effect benefit. There was some time in the past where the resilient coral benefited the resistant coral. So it gets that higher cover with the resilient coral having been around compared to it being alone. With adding external recruitment, with and without macroalgae external recruitment, we get the results I just described. Each coral benefits the other, benefiting total coral cover, that indirect positive predominating. And so really it's a competitor-enabled rescue effect that allows these corals to both benefit the persistence of each other. Looking at our two-patch model, now we have a high disturbance patch and a low disturbance patch. So in the low disturbance patch, the negative effects of competition predominate. In the high disturbance patch, the indirect positive of holding off the macroalgae predominates. So the resistant coral stays about the same but with lower variability. The resilient coral, that it's got higher cover in that low disturbance patch, so the negative effect of competition is predominating in its total coral cover. So it actually loses a bit of cover with the resistant around, but the total coral cover is higher with both together. So Finally, for competitive effects, here we have interspecific and intraspecific equal. Here we have interspecific less than intraspecific. Both of these, resistance effect on resilient is the same as resilience effect on resistant. In the lower right here, we have the resistant coral as the superior competitor. All of these have each coral having that indirect positive benefit to the other. Both coral gains from having the other around, leading to a benefit of your total coral cover. But the one exception to this indirect positive outcome comes when the resilient coral is the superior competitor. Because then the resilient coral early on drives low coral cover of the resistant coral. It's not there to benefit the resilient coral, and then it experiences a detriment as the resilient coral dies out from these high disturbances. And so this is the one world where the resistant coral declines, leading to the one case where it's better to just have the resistant coral instead of a diverse community. And so putting it all together, we can get this enhanced community level resilience from having multiple types if disturbance levels are high enough that the resilient coral would have collapsed otherwise. And if the resistant type is this equivalent or superior competitor, and our different dispersal dynamics show that the mechanism for this enhanced community level resilience is competitor-enabled rescue. So putting this all together in terms of what drives response capacity to future global climate change and ocean acidification, we see this overarching role of survivorship and population persistence and adaptive capacity. On the community level, we really see this fundamental role of diversity. But in all of this, one thing that I hope comes through is the power of sensitivity analysis. Whether the global sensitivity analysis, the first two pieces, or the last piece looking at particular sensitivity to specific parameters like competition and recruitment, that this really helps reveal the mechanism of what's going on and the drivers of potential outcomes. So in a world like coral reefs, where we are really far from having the data to model the system precisely, where we can come to concrete forecasts about what the community composition will be in 50 years in this one spot. What we can do is say, what is the relative influence of different pieces on the outcome? That's the part we can say with confidence. That's the part sensitivity analysis can inform. And that's the part that managers need to know, okay, what do we prioritize? So I would like to again thank the organizing committee for the opportunity to speak here, thank all of the people that have helped contribute to these ideas, and thank you for listening.
Very important announcements about the poster awards if you just wait till after the Q&A with Marissa. So we're happy to field questions. And remember, raise your hand, we'll bring you the mic. Any questions? She was very clear, I realize that.
I'll throw in a quick question.
Okay.
So how do macroalgae feel about ocean acidification and climate change?
So the macroalgae are not a calcifying organism, so they're not very well affected by the ocean acidification. Climate change is going to depend on the species. Some will grow faster with higher temperatures, other are more temperature sensitive. One of the things we're more worried about is crustose coralline algae is a calcifying algae that encrusts on the surface. We lump that into free space. That actually is something corals and other marine organisms like to recruit to. They release a chemical cue that says this is a good rocky substrate to recruit on, and they are going to be negatively affected by both temperatures and ocean acidification. So there is a whole group looking at that whole question of crustose coralline algae and how that then might affect recruitment of other organisms.
So the question I had is, this multi-scale theme continues to arise through all the talks, and I'm always interested in linking up of the different scales. So have you done a model where things are embedded, and so something is affecting at the molecular level, you're seeing it at the community level, for example?
We have not yet done both having the evolutionary change and the multi-species piece yet. Our jump was to connect the evolutionary change. Oh no, actually, let me correct that. We did do with the thermal tolerance model that we had two species, a massive type and a branching type, with evolution and the symbiodinium within each species, because we wanted to look at species composition versus genetic composition. And in particular, are you more worried about protecting a place that starts with high cover of your resistant type or of your massive type or your branching type to your end outcome? Or are you more worried about, well, maybe high cover signals a place with high past stress? And so that actually might be a place with higher future stress and less likely to persist into the future. And we found that that signal of high past stress outweighed community composition, that the communities, even though if these are slow-growing organisms, could grow in rapidly enough. So that initial diversity mattered less to the long-term outcome compared to what your stress level is for your location.
That's great. Are there any questions? Yes, Jonathan.
So the community stuff at the end is very cool and very suggestive, and I'm really wondering about scaling it up to the whole community and about, you seem pretty unsure about how even your two extreme types are coexisting. So what plans or thoughts do you have about community level models and about understanding coral coexistence?
Yeah, I mean, so it's interesting because in the 90s, there was a lot on what the heck drives the coexistence of all of these. And there were some really neat meta-community models trying to look at, is it a competition colonization trade-off, what might actually be driving that? And I mentioned that whole stinging cell thing, and there was all of, I think it was Judy Lang did all this really great work on can we put together competitive hierarchies of these species. That was in the 80s, and that's been quiet. And I've been really surprised by that, and I think there's a real opportunity to dig back into that world. If we can understand what's led to the diversity to today, that can help us think about what might continue to lead that into the future. One of the more interesting recent studies is a Darling et al. Study that categorized corals, did kind of the Grime et al. Grime did this for plants, right, back in the 70s, of do we categorize, there's the resistant type, the resilient type, there's kind of a weedy type as a third category. And they found that there were those three plus a fourth broad category that cropped up sometimes. So I think a next step is to start to use those categories and better map life histories to those as an intermediate until we get to the 200 species world.
So thanks, great talk. So kind of following up on that question, so this model and this approach I could see being applied to corals and plants and other kinds of species where they interact and they compete for space. Mm-hmm. Could you make a wild extrapolation and think about how this might be able to be worked for species which don't really compete for space, but they compete for other kinds of resources? So maybe ungulates in a boreal forest, or that kind of thing. Could you expand the work to look at that kind of question?
I mean, I think anywhere where you have these types of life history trade-offs and response diversity trade-offs, right? That when we were looking at, so that's how we categorize this idea, this resistant and resilient type, the response diversity. When we're digging into literature, there's actually some interesting studies into, I think it was bird systems that had the response diversity. And I think that it's very common to have this variety of life histories. You're slow-growing, long-lived organisms that can kind of wither the change, versus your faster-growing, shorter-lived organisms that track the change more and bounce around. And so when you have a very disturbance-driven community, that's the real question. Is your global change happening through increasing disturbance levels? Then it's when that response diversity is really going to matter. Versus is the global change happening as a more gradual ramping up? Then I think other types of diversity are going to matter more than your response diversity.
Well, given the time, let's thank our speaker for a fantastic talk.